Okay, friends, now let's start to talk about again the practical purpose of bioinformatics, and we are solving the questions, and we have solved up to this part, which is the prediction of the secondary structure of human prion protein. We have seen the structure predicted, and we have seen the different regions of the different domains of the protein, like alpha helix, beta sheets, and also the conserved domain. And this prediction mode, what kind of mode is also selected, we have seen. But now let us look at the retrieval of the 3D structure of this human prion protein from PDB or protein data bank. Because no, that protein data bank is the structural protein database, because we have seen the introductory video that there are database variations. There are databases called sequence database, like PIR is a protein sequence database, SwissProt is a protein sequence database, UniProt is a protein sequence database, but PDB is a protein structure database, so as cath and scope. So we'll be looking at PDB, which is the most popular protein structure database, and from this structure database we'll download the 3D structure of human prion protein depending upon our need, and we'll be seeing the options, and also then we'll open this file in a 3D structure viewer, which is in this case we are going to use the Jmol. There are also other viewers like PyMol or Rasmol, but in this case we'll be looking at the Jmol. Okay, so let's start. So simply we write PDB or protein data bank. So simply by clicking this protein data bank, it will take us to the protein data bank uh, site. So this is the protein data bank site. It will take some time because other jobs are running in my browser. So it is the protein data bank. Now this is the front page of the protein data bank. It is available for iPhone application also. You can look at it. Anyways, now what we can do here, we can search for a protein name, and we can search by protein author name or also the molecule name and everything. So let's start to search. So we can write the human prion protein. Remember, we can search it by typing human prion protein. Whenever we search this human prion protein, it is giving us the list of organisms and also list of phylogenetic tree. Now the list of organisms we are getting it of Homo sapiens. If we click on the Homo sapiens, it will take us to the Homo sapiens part. So simply clicking the search menu, it will take us to the protein of human prion protein here. Okay, so this is the result of human prion protein. Now this is a huge result because this protein data bank, it is huge resource of protein structures. So you get a large result. Now you can see in Homo sapiens we get 24,104 different type of proteins. In E. coli we found it 4,793 proteins. A similar type in mass molluscas we found 4,147 proteins, and so on, because it highlights only Homo sapiens proteins, because we want the prion protein of Homo sapiens or human, right? That's why it's highlight the human or Homo sapiens, and fade other proteins here. Okay, now here what we can find in the next thing, we are having the query refinements. Here we are having the query refinements depending upon the organism. Like Homo sapiens, there are 58 query match. In Mass mollusca 7 match. In Bos taurus 5 matches, and so on. So we want the protein of Homo sapiens. If we click this one, it will take us to only the proteins, or the prion proteins of Homo sapiens. We refine our search, so now it comes down to 50 only, right? Now among this we are also having different parameters, like parameters of X-ray resolution, release date, and say polymer type, say protein, or different things is only proteins are available. Now what are this Excel resolution is telling us, because whatever we're looking at is this PDB or protein data bank, these are real structures. These are real structure. This is not like Fire 2 or Swiss model, because in Fire 2 or Swiss model what we have seen, the prediction of the 2D model or the prediction of the 3D model, but in this case we are getting the real 3D image, which is captured using the X-ray diffraction, and also they are captured utilizing NMR spectroscopy, for example. So these things are important, because here what we are getting is the live things, the real things. So we can get so scientists have researched with these prion proteins, and they have studied it using X-ray diffraction crystallography. Now this X-ray crystallography results can give us several different resolution. The resolution can be of less than 1.5 angstrom. It can be a better resolution like 1.5 to 2 angstrom resolution, can be 2 to 2.5 angstrom resolution. Now we have the better resolution, which is less than 1.5 angstrom resolution. So it's only three molecules, such molecules are available, and the molecules that are between 1.5 to 2.0 angstrom resolution of X-ray crystallography, six such molecules are available. Now we are trying to find this type of molecules which are having the resolution of 1.5 to 2. Then we click this link, it will take us to only those proteins, only those prion proteins having the resolution of X-ray crystallography with 1.5 to 2 angstrom. Now we get our desired results at the bottom. So you can find these are the results. These are the prion protein results, human prion proteins, and we are having the structure is discovered by the X-ray diffraction with the resolution of 1.0 angstrom. Because we have selected the resolution scheme from 1.5 to 2, this one is having a better resolution 1.25 angstrom. This one is having uh, 1.80 angstrom, and so on. So what we will be doing here, we will be focusing on say this one. So this is a segment of human prion protein. So you can see the segmentized portion. So it's a segment of prion protein, because we know that human prion protein is having 253 amino acid sequence, but this protein is having only 138 to 143 sequence with it. So the structure they provided is only from 138 to 143 amino acid sequence. But we won't want this segmentalized portion. It will be good if we get more residues. So here it is a human prion protein variant, which is a 103 nucleotide sequence long. This is another variant, it is 284 sequence long, and this is also another having 284 sequence long, having 1.80 angstrom of X-ray diffraction study. So two compounds are there, one polymer, one ligand. We won't want these things actually. So let's go back to our search and refine our search again. Remember, we have got so many searches here. So this is the NMR structure of prion protein mutant. So say these are the different fragment of human prion protein. So these are the fragments. You can see only 70. So here the residue count is telling you how many amino acid sequence they are able to capture, and they're able to make a 3D structure. This is only 72. You won't require that simple small amount. Here it is a NMR structure of human prion protein. It's having 146 residues, and this is a NMR structure. So let's take this one, because these are the filters we can apply, details we can apply. We don't require them, so we simply take up this one. So click on to this particular protein, which is having 146 amino acid sequence, and it is the structure is placed using NMR spectroscopy. Now here are the details of the scientists who have worked with this, and this is the journal. It is published in Cell from 248 to 254 page of the Cell, and this is the PubMed abstract of that. And we can see here this is a graphical representation of prion proteins and pure-like proteins. It is 146 amino acid long. Length is given. Mutation, it is denoted with V209M. This is the type of mutation. This accession, these numbers are telling us what type of mutations are done. This is the gene names PRNP, PRIP, and PRP, short form of the gene name, and that's it. So if we look at the right hand side, we get the structure of our protein. Now here is our protein structure. Now we can view this protein using simple viewer. We can look at via the protein workshop or kiosk viewer. Now we can also download the structured data. Okay, we can also download the structured data. So we can download the file is in PDB text format, PDB file, and all these things. So we simply click here pdb file bracket text to download this structure of the protein as a pdb file. So let's download it, save it. So we have saved this file. Okay, so this is our protein. Okay, now if we click onto this image, it will take us to the view of the picture. So this is the 3D structure. This is actually not actually 3D, it's a 2D representation of the structure of a prion protein. It is having the alpha helical region, 3 helix, and rest of the part is loops and coils. No beta strands is shown here. Okay, so here it is, the first tab whatever we are watching, it is the summary tab. Now if we look at 3D view, it will take us to the next tab providing the 3D view of our protein. Provided you must have the Java applet in your browser, the Java enabled in your browser, otherwise you cannot visualize this. In any kind of bioinformatics study, you must have fully updated server as well as the Java system to visualize these molecular structures. So let's move in here, say this telling us, so let us allow this, click allow to start this platform. Yes, now Java is loading, and here we can visualize the 3D structure. Now if you don't have the 3D structure viewer in your computer, you can visualize this in this website, but you need to have the Java enabled. But if you are having a 3D viewer in your computer, then you can download the structure in PDB text format, then you can visualize the structure in your PC also. We are going to see also how to visualize the structure in your computer using Jmol software. You can also visualize it with Rasmol or Pymol softwares. Now here it is giving the structural details. Say structure is PDB entry solution in NMR structure, global symmetry, and this is a front view. So you can change this view to left, back, right, and all this. And here is the secondary structure that we are viewing. We can look at symmetry, we can look at custom view and all these things. Now let's say I accept and click run so that the picture is allowed. So again, the online 3D viewer they are using is also Jmol. So here it is. This is the ball and stick model of the 3D structure that we are visualizing. As you can see, this is the ball and stick structure with having blue color, red color, and other colors. So you simply click on here, and we can go to style. And by going to style, we can change their styles depending upon our need. Here, say the style is changed to the ribbon diagram. So okay, so you can look at it. Now if you select style again, you can go to atoms, and depending upon atom you can change the style, or you can go to structures, and here you can, in the structure we can select ribbon diagram or cartoon diagram or backbone diagram. So if you select the cartoon diagram, this is a cartoon diagram. If we go to style, and among these structures we can also go for ribbon diagram. This one is a ribbon diagram. You can see it's like looking like a ribbon. Now again we can go for style. You can change the structure again for say backbone. This is the backbone diagram only, so you can change depending upon your need. You can look for the van der Waals interaction, so you can go for uh, the 3D view also. You can go for schematic diagram, say the ball and stick model, then water wireframe model, cartoon model. So if you click ball and stick, it will convert it to the ball and stick model. Okay, we can zoom in there, say this is the zooming in. We can zoom out also, say 50%, it is pretty small now. Say zoom 100%, it's nice. Then say spin, you can click on, so it will keep on spinning. This is the front. You click back left, it will spin left direction, so only left will be seen. If you click right, if you click back, this is the back part. If you click right, this is the right side of the protein structure, it will be shown. Measurements can be done. So how can you measure these things? There are scales also for the measurements. Double click. So let's say we will measure from this yellow till this one. So from this one, say this one, it is giving us the measurements. As you can see, this is telling the distance between these two atoms is 22.32 angstrom. Simply by clicking twice and then click here, it will give us say Here you can look at the distance scale. This is the red color thing is the scale. It is attached to this blue color thing. Now if we click it here at this red, it will give us, say, from here, this is the scale. If we click at this bottom, it will give us the distance. So let's say from the top we create this, at the bottom we are getting the distance. So the distance they are providing us here, as you can see, 28.5 angstroms and so on. As it is moving, the distance is changing because this is the distance scale. Again click here, so it will provide us the distance value. Okay, so we can do many things with them. So now we can uh, go for, we can off the spin, we can change the color, we can change the color according to their uh, atoms, say by scheme. So this coloration is done by scheme. Also we can change the color, say the atoms, all the atoms are black, so now everything will turn into black. Right, now if we go to the color again, all the atoms colors say orange, all the atom color will be orange now. So we can change different uh, colors and everything according to our need. This is a cool thing about Jmol. We'll be studying Jmol in our future case when we are doing the structure study in our PC. So that's it. You can download it, and we have downloaded this file already simply by dropping it, clicking this drop down menu from this download tab, download file, and just save it in the pdb text file, okay, for the future use. So that's it, and I hope this video is helping you, and many things to remain. So if you click on sequence, it will take you to the sequence of this 3D structure of a protein. So the sequence of the protein will be provided to you. Say this is the sequence, this is the graphical representation of the sequence, telling you that this red colored segment region is alpha helix, this yellow segment is a beta sheet, and this purple segment is a turn of your interest, and the rest of the part, this black colored region, is something they haven't identified yet. So this is the structure of their protein, and this is from the uniprot. So the sequence, remember the sequence server for protein is uniprot, so they get this sequence from the uniprot. So if you click on this selection, add annotation, you can simply click this drop down and you can select this domain parser. It will take us to the domain parser segment. Only we can see the domains here in this green color segment. If we simply click here as normal, or previous view, it will take us to a simple view of that. Okay, so again, if we look at the annotations for this sequence, it will take us to the annotation page. Here is the annotation. So the comment of this protein, the prion protein is thought to be the infectious agent that causes transmissible spongiform encephalopathy and all these things. It is given different things. So type is a domain. The domain is called prion dopel alpha helix domain. The domain is identified, and you can look at it. We can look for sequence similarity tab for the similarity of this sequence with other sequences. So we can see the sequence similarity with the cluster number. Here is the 100% similarity, 95% similarity, say with this number. So if we click this number, we can see the clusters with which they are having 98% similarity. So these are the accession numbers with which they are having 98% similarities. If we click the download button, by selecting all of this, the structure of these proteins also be downloaded. Okay, we can go for 3D similarity, where we need to look for the 3D structure similarity. Again, like the sequence similarity, we can also find the 3D structure similarities in this tab. Okay, in this literature we can find from where we've taken all these things, and methods. Inside the methods we can find what method is utilized for tracking the structure of this molecule. Here says the method is NMR, remember, The NMR method is taken and every detail is given for the NMR. Solvent system utilized is 95% H2O and 5% D2O. Ionic strength was 10, pH was 4.6, and temperature is 299 Kelvin, and everything is provided at this place, right? The method is stimulated annealing. Molecular dynamics is the model, and here say different IDs and the equipments that we don't need to know now. If we click the geometry, it will take us to the NMR result of the geometry result of this protein. uh, this is pretty hard time for us to understand this concept because we don't require them, because it is giving us different chain and the data coming from NMR. We don't require these things because we are at the pretty beginning, but we can search proteins like that depending upon what techniques they used for cracking the structure of the protein, and also depending upon the name of the author and also the type of protein and everything. So that's it, and I hope this video is helping you. Thank you.